Hello, and welcome to your first fully vodcasted lecture of the semester. This will likely be the first of what will be maybe 3 or 4 full vodcasts, so you can be looking forward to that unique way of lecture presentation and maybe having some other fun ways to engage in the material via this virtual mode. Let's start with a few announcements. Most of these, in fact, all of these are review. I think that most of you by now have sent me a topic for your poster presentations. If you're still working on those, or if you're still bouncing ideas around, just let me know and we'll try to get those finalized as soon as possible. Again, your exam number 1 is going to be on Tuesday, and that's the 18th. I will be offering reviews for that, and it looks like I've managed to make another typo there. This is September the 17th that the review will be held the Monday prior to your exam. On September the 18th, I will hold a review. In fact, I will hold 2 reviews, one from 3 to 4 p.m. And if there is no one coming into our regularly scheduled lecture, we'll hold it right after that in the same room. If we need to, we'll proceed upstairs to the lab and we'll do the review there. We will also do another virtual review, 7:30 to 8:30 p.m. on Illuminate, as I have mentioned. And I have already talked to you a little bit about the format of the exam. Just a quick review on that. 25 multiple choice questions. Look to your homework for the type of questions to expect for the multiple choice. In terms of question numbers 26, that is the epic question where you might want your colored pencils and whatnot, because you'll be drawing for me a gram-positive and gram-negative cell wall. So that's a guaranteed question. You're all going to have to answer that one. However, after that, it becomes freedom of choice, because at that point, for questions 27 through 32, you choose to answer only 4 of those questions. Please circle those that you choose. If you've been embarking in the discussion of omnivore's dilemma and interacting with your peers on that discussion, then you'll choose to answer that question and won't need to answer another one. You'll have only 3 questions to answer rather than 4. There will be an SI session today from 3 to 4 p.m. as usual in the lab, so if you're needing any help at all with your homework or just have basic questions, that's a great time to talk with Callie and Marielle. Lab, of course, we do have the gram stain report coming up on Thursday the 20th, and this is the point where I will stop announcing pre-labs, because by now you're starting to get into that habit of submitting the pre-labs every time you have a lab. Let's jump into some really riveting content today. I know you guys are jazzed, probably more jazzed than I am, although I don't know if that's possible, about talking about the layers external to the cell wall. Remember how we ended off our coverage last time really looking at the depths of different kinds of bacterial cell walls. And we made note that sometimes that's not the most outer of the outer layers of a cell. Sometimes they have another layer, another protective layer, or another functional layer. So I'd like to start by talking about one of these types of layers that is collectively called the glycocalyces. The glycocalyx is a layer outside the cell wall that is generally comprised of carbohydrate, polysaccharide. So we recognize that as potentially having a very protective role, because remember how water-loving carbohydrates are, and hydrophobic toxins that might hurt a cell aren't going to be very likely to pass through a blanket of this very water-loving polysaccharide layer. So things like viruses and hydrophobic toxins will be excluded by the glycocalyx. And also, if you think about how sticky this might make a cell having this outer carb layer, it allows cells to adhere to surfaces. So if you run your tongue across your teeth, you'll recognize that you're touching a little bit of glycocalyx, because Streptococcus mutans, one of the lactic acid bacteria in your mouth, adheres to your tooth enamel using an outer capsule that allows it to really hold tight to your teeth. So in protection, in attachment, these are some of the roles where we see glycocalyx really getting involved. We did talk in lab about the capsule, and we actually even stained that capsule in Klebsiella pneumoniae, and we talked about some of the more distinctive and unique functions that a capsule can have, including protection in phagocytosis. So remember, these capsules make cells really slippery little suckers, and so when a engulfing macrophage, for example, tries to engulf an encapsulated bacterium, it's going to slip right out of the grip of that macrophage. There are also, and we didn't talk so much about this in lab, something called a slime layer. A slime layer, unlike a capsule, is much less well organized. It's much more diffuse, it's much more irregular, and it's also much harder to observe. So in vitro, that is on top of your lab bench, it's difficult to look for slime layers, but we know that in vivo in the body, they often exist. For example, the pathogen Staphylococcus aureus is thought in vivo to be covered in a slime layer that's very protective to it, but it's hard to see that often when we observe it under a microscope. There is another layer that can be very different from a glycocalyx, and functionally it serves many of the same roles, but rather than being comprised of just this polysaccharide, this sort of diverse polysaccharide, it actually tends to be composed of a very distinctive structured layer of protein or glycoprotein. Now, the thing that makes an S layer very, very unique is the way in which the structure is distinctively arranged. That is, it tends to look like floor tiles. So I'm going to make a quick drawing where we often see S layers having this sort of regularly repeating structure, maybe looking like floor tiles and forming maybe like this one. I'm showing that these floor tiles are kind of like hexagons. So remember, these are protein or glycoprotein. My analogy to floor tiles is simply to help you visualize how they would regularly repeat. And because of that repetitive structure, it makes for a very discrete, very organized layer with very uniform pores in between the protein and glycoprotein. And I thought this might particularly interest Sakun because I have an article about how these can be used in a sort of industrial manner. That is, these can be used to make these layers that have very distinct pore sizes, and the pore sizes can be used as a membrane, essentially allowing only things of a certain pore size to pass through them. So I'm going to read to you just a little bit from this article. The name of the article is Biotechnology and Biomimetic with Crystalline Bacterial Cell Surface Layers, or S layers. And what it says is essentially S layers have broad application potential in biotechnology, including functioning as biomimetic membranes, because they have pore sizes that are fairly uniform between 2 and 8 nanometers. So we see these S layers sometimes from particular bacteria. One particular bacterium that tends to form them is called Caulobacter crescentus, which we'll talk more about that bacterium. It's wicked cool. But one of the ways in which it's used in these sort of biomimetic ways or in biotechnology is by using the S layers as high-density monolayers and artificial surfaces and maybe artificial membranes. So a lot of potential in biotechnology for S layers. So I thought that was pretty cool. But that's about us and how we are tapping into something that bacteria naturally make. But what about the bacteria? What is the reason that they're making an S layer? Once again, like glycocalyces, this is very protective. It may protect against pH fluctuations, osmotic stress. It may protect against toxins and enzymes in the environment that might be a threat to the cell. It protects certainly against phagocytosis, predaceous bacteria. We'll talk later about a mad killer bacterium called Delovibrio that bores its way into its prey bacterial cells. So a cell that has an S layer is quite glad that it does, because it will be less likely to be prey to Delovibrio. So it may help to maintain shape, rigidity, promote cell adhesion, just the way that we talked about a capsule doing, and certainly protect against phagocytosis. So many, many functional roles for an S layer, and we might say that both S layers and glycocalyces. So looking at our drawing down here that sort of summarizes these, S layers and glycocalyces have some of the same functional roles, but one of the main things that differentiates them is overall composition, overall morphology. In the S layer, we see the really floor tile-like patterns that are always made up of protein or glycoprotein. In glycocalyces, we tend to see them being made of polysaccharide, occasionally glycoprotein, but we don't see that distinctive floor tile-like pattern. Glycocalyces, further, as we mentioned, can be distinguished as being either capsules or slime layers, and slime layers are very diffuse and irregular and hard to observe. With that review in mind, let's talk about the very last type of layer external to the cell wall that we'll hit on, called a sheath. And a sheath is wicked cool because essentially what it is like a straw-like structure or a tube that surrounds a community of cells. So if you visualize it as being something like this, where you've got this sort of straw-like structure, and then you've got cells that line up inside of this, fairly close to one another, and form this sort of community that sits inside of this straw-like structure. You could see how something like that would be incredibly beneficial if you happen to be an aquatic bacterium. A sheath allows you to adhere to a surface and live in a community and to hold on while maybe a river is flowing by and you can catch nutrients. It's a protective structure. It's a structure that allows you to set up a community of cells and to get nutrients, right? Have great, efficient nutrient acquisition. So let's just jot down, we'll talk more later about sheaths, that they are a tube that surrounds and holds a linear chain of cells together. So this summarizes our layers external to the cell wall in terms of these protective layers that sit outside of the cell wall. I also, though, want to talk about something that we might consider external to the cell wall in our structure that allows for motility, that is the flagellum. So maybe my favorite structure of the prokaryotic cell, the flagellum moves like a propeller. It actually can move in one of two directions. Either it can move clockwise or counterclockwise. So you're essentially getting that motion as you would think of it as being like a propeller, propeller type motion. So let's talk a little bit about flagella. Flagella plural, flagellum singular. And some bacteria have one, some bacteria have many. These are long, hair-like protein structures. We recognize them as having a very long projection, and in this picture down below, I've only showed a tiny piece of the long, hair-like projection, and I'm focusing a little more on the way in which the flagellum inserts into the cell wall of a bacterium. Now take a moment to look at the cell wall of this bacterium with its outer membrane, its periplasmic space, and its inner or cytoplasmic membrane. And I imagine you're probably already yelling at the screen, I can kind of hear you, saying this is a gram-negative bacterium, right? We can tell that by the structure. So this is how a gram-negative flagellum would be structured and inserted into the cell wall of the bacterium. So we mentioned already that these flagella will spin like propellers, and they do it fast. In fact, if you take something like Vibrio alginolyticus, this is a bacterium that can spin its flagellum at 1,000 revolutions per second. It is so quick. I mean, in fact, let's take an example, right? We know Jacob likes to run a lot. So Jacob, if you were out running and you managed to go 82 miles per hour, now not to discount your speed or anything, but not going to happen, right? So these bacteria can move at what would be an equivalent for you of 82 miles per hour. That's wicked fast. But don't worry, you have something up on a bacterium, and that is that you actually know how to move in a thoughtful, spatial sort of direction. That is, you can look at one point and you can say, I'm going to run from this point to that point. A bacterium can't do that. So it better be able to move fast because it never takes a direct route anywhere. But it is wicked quick, right? In fact, I did a couple of quick calculations and found that this is still far faster than what we see our fastest land animals moving. So this is a really, really quick speeds. Even E. coli that's not as quick as Vibrio alginolyticus can move very, very rapidly. So basically we see these very quick moving flagella, but that's not the only role that they may play. Obviously, they're playing that role in motility and getting around, but they can also be virulence factors. And I have with me today one of my very favorite stuffed microbes. I have a large collection of stuffed microbes. They're wicked cool. Come see them sometime in my office. But one of my favorites is my bacterium called Helicobacter pylori. Helicobacter pylori has what we call Lophotrichus flagella, and you can see they're kind of shown like here, but really what Lophotrichus means is that these flagella literally are a tuft of flagella at one end of the cell. Well, turns out that Helicobacter pylori is able to spin these at very fast speeds, and it uses them as a virulence factor. Helicobacter pylori, you guessed it, is the one that causes most cases of peptic ulcers. So it uses its its flagella to bore into the stomach epithelial lining and to cause some of those symptoms that come along with ulcers. So you'll notice that some of my students, when I pass my Helicobacter around, did make, in fact, a gangster headband for my Helicobacter pylori. A scarf too, oddly. So let's go ahead and look at the parts of a flagellum. And I just want to take a quick note and label some of these parts. Up here, this sort of long hair-like projection, this is called a filament. Here, this section is called a hook. And you can see how the morphology of the hook is what makes it so that a bacterial flagellum can only rotate either clockwise or counterclockwise, right? It can only go one direction or the other. Down here, this whole region that inserts into the cell wall is called the basal body. Now, the basal body has a couple of different rings in it. It has an L ring, a P ring, it's also got an MS and a C ring. So this is the MS and C ring. Now, you saw me glancing at my notes there because I'm just trying to get used to this. This is new nomenclature. And in fact, it used to be that these were the S and M rings. And I'm pretty sure that they stopped naming them that because I used to make dirty jokes about it in lecture. But in any case, we now understand the parts of a bacterial flagellum and how those parts are inserted into a gram-negative cell wall. Don't worry about the gram-positive structure. I just wanted to highlight some of the structures that we see in a bacterial flagellum and how it works. Let's look at the ways in which flagella can be distributed on the surface of a bacterial cell. We mentioned Lophotrichus already, but there's also other types of orientations that we can see. So let's look at that next. Some bacteria only have one flagellum. We call these monotrichous. For example, those of you who are thinking that your unknown bacterium might be Pseudomonas, and if you're wondering if that might be you, you might have had short little gram-negative rods, and if you found the dichotomous key that's online under additional information on the lab website, you might be starting to think, well, maybe I have Pseudomonas. Now, we're going to talk a lot about that awesome bacterium coming up. But in fact, it does have only a single flagellum that comes from one side of the cell. We call it monotrichous, but we also call it polar because it's emanating from just one side of the cell. Some bacteria have flagella that come from both sides of the cell. We call these amphitrichous. So if you picture that some bacteria could literally have another flagellum emanating from this side of the cell, that would be amphitrichous. And in lab, you saw the demo scope that had a long sort of spiral-shaped bacterium. It kind of looked like this, where you had this long spiral-shaped bacterium. And then that bacterium further had amphitrichous flagella, so emanating from both sides of the cell, we saw flagella. So that was a crazy looking one, right? It was an amphitrichous spiralar cell. That's a tongue twister. So the other one that we can see, we talked about Lophotrichus. And, Halloween's coming pretty soon. This makes a great Halloween costume. All you have to have is a little bit of a bacterial hat, and then you can have your Lophotrichus flagella coming out of the top, and you just feel like you have a great costume, right? So we also can see, some of you have an unknown, that if it's motile, it is motile via peritrichous flagella, meaning that the flagella literally cover and coat the whole surface of the cell. Some strains of E. coli have this type of motility. In fact, let me grab my other stuffed microbe. I've got an E. coli up here. And they didn't do a great job because they tried to make the peritrichous flagella look like hair. But in fact, E. coli would be covered fully with flagella if it was motile, distributed all over the entire surface of the cell. Marvelous. So we can think then about how would these bacteria move with these different kinds of flagella, and what would cause them to move, right? What would inspire a bacterium, to God to move it, right? Well, obviously it's not fitness, but instead it is getting somewhere, right, to something. And so these flagella will move when needed to move to a resource or away from some sort of repellent. And they do so in these two different ways, right? They either move clockwise or they move counterclockwise. If they move, sorry, you should write down entire cell surface. Maybe you already got that down. If they do move in a counterclockwise direction, then we're going to recognize them moving in a run. That is, they're going to go in one direction in a run. But if they move in a clockwise direction, then they're going to basically stop and undergo what's called a tumble, which allows them to make direction changes. So Jacob, this is why I said that they don't move in the same way you do, right? Rather than going from point to point and saying, I want to be at that point and instead of this point, they move all of the time. That would be good for our fitness, wouldn't it? And they temporally sense. That is, they sense where they were before, where they are now. They tumble again, where am I now? Move again, where am I now? Move again, where am I now? Right? They're sensing each time they move and each time they tumble whether there's been an environmental change. So let's write a bit of that down. Again, counterclockwise motion is going to move them in a run. They're going to head in a straight line for a little while. Versus clockwise motion is going to move them in a tumble. This is going to be a direction change. So they'll sense the environment when they do a tumble, and if it has changed, then they might lengthen their run. The change, of course, will come from something in the environment. It might be that it's a chemoattractant, or as Levi will tell us, it could be that it's light, right? It could be that the bacterium is phototactic and it's moving in response to light. Whatever the environmental cue it's looking for, each time it tumbles, it will look for a change in that, and that might dictate a change in the runs. So this movement in response to either a chemical or a light or some sort of environmental cue. Often it is an attractant that is a nutrient or a repellent that is a toxic byproduct or waste product of the cell. So they may be moving away from repellents and towards attractants. Now they sense both and they'll compete for these sensing fillings, right, of moving away from a repellent and towards an attractant. And you might be thinking, well, how do they choose? Right? How do they choose whether they're going to go towards an attractant? Say there's also a repellent there in the same space as the attractant. What trumps the other one? Well, they do compare chemical concentrations, but it turns out that bacteria are more sensitive, more well-honed to an attractant than they are to a repellent. In fact, there are double the number of receptors for attractant than there are for repellents. And bacteria can respond to attractants at a concentration of like 10 to the minus 8th molar. So they respond to attractants at much lower concentrations than they do to repellents. So you can see how the tug of war occurs there, and eventually they make their way in one direction or another. But as I mentioned, they do not do so spatially the way that we do. Instead, they do so temporally. So they sense one environment, move again, sense that environment, move again, sense that environment, move again. You get the point. And eventually they make their way to the point that they're going. In the absence of a chemical attractant or repellent, they still move. They move in a random way. That is, they'll make a run, they'll make a tumble. And I'm going to kind of highlight that down here. They make a run, here they make a tumble, here they make a run, right? And they basically move in a random way unless there is some sort of chemical attractant or repellent that causes them to move in a different way. So the chemical attractants and repellents will bind to receptors called chemoreceptors. These chemoreceptors are found on the surface of the cytoplasmic membrane, and the receptors send a signal to control the movement of flagella. Now, I have a feeling that Schwa is not buying this, and he's saying, Rachel, there's a lot more detail to all of that than what you're telling me. You are spot on. In fact, there is a lot more detail. There's a couple of receptors that are going to be those receptors bound by these chemical ligands. These receptors lead to entire basically domino effect of phosphorylation and methylation events. Eventually you see an overarching change that's going to allow, there's a flea type influx protein that allows protons to swoosh in. And remember how we said the PMF can power a lot of things? Well, the PMF is what's opening the floodgates, or passing in through the floodgates and allowing for the flagellar rotation. So there's an entire set of apparatus that surround every basal body of a flagellum that allows for influx of protons and resulting movement of flagella. See me for more details if you'd like. So in the absence of a chemical gradient, there are equal numbers of tumbles and runs, and the bacterium moves randomly. And that's what we see in this figure here. We see the random numbers of tumbles and runs basically leading a bacterium nowhere, but it moves all the time because that's the way that it senses its environment, is by continuing to move. Suddenly, if there is a chemoattractant or a chemorepellent in that environment, the cell will start to lengthen its runs and lessen its tumbles. And in a method that we call a biased random walk, it will eventually make its way to where it wants to go. By no means is it a direct route. So when there is a chemical attractant, the length of the run increases and the bacterium eventually moves towards that attractant. This graph here is basically showing you what happens when the runs become elongated. When each time a bacterium tumbles, it senses that it's coming closer to an attractant. So the gradient in color here, where it's darker green, shows you the gradient in the chemical attractant, getting greater and greater until you get to the darkest green region in that picture. So this biased random walk towards a chemical attractant. Let's end our coverage for the exam and also end our coverage of prokaryotic cellular structure and function by looking at something a little bit smaller than bacterial flagella. And in fact, in this picture that you see down here, we can see bacterial flagella right here. So these that I'm trying to outline in blue there are flagella. But much smaller than flagella are something that are sometimes termed Fimbriae, and you can see them projecting out here from the cell. Some textbooks also call Fimbriae by the name of pili. Our textbook calls everything Fimbriae unless they're referring to sex pili. Remember those structures that allow bacteria to pass plasmids from one to the other? That's a sex pilus. Our book reserves the term pili to describe those, rather than using pili as a generic term to describe all of these smaller surface projections. But just like flagella, Fimbriae are also hollow cored. They're not made up of the flagellin protein, but instead they have a little bit different composition and are shorter and thinner than flagella. But they do have the protein subunits, just as a flagellum did, that are arranged helically in order to create a hollow cored structure. So they share that element of structure with a flagellum, but they're just much, much smaller and thinner. They can allow for a couple of different things. One of the things is adherence to specific surfaces. For example, certain bacteria that cause urinary tract infections, they utilize their surface Fimbriae and adhesin molecules on those to bind to specific host glycoproteins that line the urinary tract. And thus these bacteria hold on tight. It doesn't matter if urine is rushing by. They're like, woohoo, and they hold on to whatever surface they're adhered to, thus increasing their virulence and allowing them to be more likely to cause infection. Now, environmentally, we also see pili or Fimbriae playing an important role in motility. We have some types of bacteria that move by a really weird mechanism called swarming motility. And swarming motility is basically like these short quivering movements, and there's a group of bacterium called the myxobacteria that do this. In fact, there's one of our faculty in our department study these. They move by these sort of twitching motions across solid media. And sometimes people term them wolf packs because they're working together and moving together in this sort of unusual sort of motion. So very cool function of Fimbriae in terms of swarming motility. And then lastly, pili, as we mentioned, is the term that your book reserves for sex pili. These are the structures that are formed If a cell has a conjugative plasmid, has a plasmid that encodes for its own replication and motion, that is, across from one donor cell to recipient cell. We'll talk more about the details of sex pili later, but suffice it to say, right, those are those nightmare structures that allow cells, and I shouldn't always call them that, because really, in fact, they're allowing for bacterial diversity, aren't they? And they're allowing for life. So in that way, we should acknowledge that. But they also are allowing for the transfer of genes that sometimes cause problems for us, because those might be those genes that are involved in resistances. Fantastic. This ends our coverage for exam 1, and it ends our coverage of bacterial cell structure and function. Gear up so we can talk about the eukaryotic cell now.